If you've had a chance to look at one of my first 4 videos, you'll notice a very big difference between them and this one. Now I have a fairly decent sized whiteboard and my website is up top. That'll make it a lot easier. Video number 1 started with this diagram. And in video number 1 we dealt with this part of the digestive system where food went through the mouth and down the esophagus. The second video dealt with what went on in the stomach. The third one considered the digestive actions in the duodenum. The fourth one, the pancreas and the liver. This one focuses on absorption and defecation. These 2 functions occur in this part. The ileum, the last part of the small intestine, and defecation occurs through the anus here. We have to prepare the material for defecation in order to get there, however. In order to get started, I want to bring your minds back to what we had at the end of the duodenum. All of the digestive actions occurred in here, and material was able to be moved again by peristalsis along through the duodenum and out to the jejunum, which is more of a transition area of the small intestine. It's the middle portion of the small intestine, and then from the jejunum to the ileum. The ileum is by far the largest part of the small intestine and by far the most specialized part of the small intestine in terms of structure anyway. When we considered the material that entered the duodenum, we had acid chyme. And it had a very low pH, a pH of around 2. The material now no longer has that low pH. You can't call it acid chyme anymore. There's actually a new name for it. The material is now called chyme. It's just still a running digestive material, but it's not acidic anymore. pH of about 8.5, 8.3. It's still about that same pH, and it carries on down here. Probably the pH gets low, a little bit lower, closer to 7 by the time it gets down towards the ileum. So the ileum itself is a very specialized structure. And if I take the same sort of anatomy and turn it this way, if we look at the ileum this way now, remember there's 4 layers to all of these tubes. The inner layer is the lumen. The ileum is not smooth like the duodenum might be, and the jejunum might be smooth on the inside. The ileum's inside surface looks like this. Huge surface area. It's made up of a whole bunch of finger-like projections that stick into the lumen. Each one of those finger-like projections is called a villus. Or plural, they're called villi. And if we take a close look at one of them, it could look more like this. Well, I think it would look better like this if I show it to you this way. One of the finger-like projections, remember it's the inside interior. This is the lumen here. This area here is all lumen. There'd be another villus sticking down here, another set of them here. They're all over the place, all around inside the lumen. And the material, the chyme material, passes over the surface of all these villi. These are intestinal glands down here. They secrete mostly water. You still need a lot of water. The villus structure is very, very unique. Epithelial cells around the outside. These epithelial cells are actually columnar epithelial cells. They're elongate cells, as I tried to indicate with this picture there. They're elongate cells, but the exterior surface of them even has more surface area. So where one of these things is called a villus, these things are called microvilli. Because it provides even an additional layer or level of surface area. It has, when you look at it under a microscope, it sort of looks like a brush effect, so it's called a brush border. So the inside of the ileum has all these villi in it, and they are all extended in terms of surface area with these brush borders. So absorption has to go through surfaces. When we started out, we dealt with food materials that couldn't be absorbed because they were so big. Well, now we're talking about things that get absorbed at this level, the individual types of molecules. The types of molecules that get absorbed, we'll just go back and mention them here again. We have monosaccharides. We'll just talk about glucose, but it's a monosaccharide. We have amino acids. Fatty acids, glycerol. We have the nitrogen bases from the nucleic acid digestion. We have phosphate groups, phosphate ions really. And we have the sugars from the nucleotides. And that's either deoxyribose or ribose sugar. So these are the individual types of molecules that get absorbed. And they get absorbed through this brush border surface here, through the microvilli. They pass through these cells into the interior of this villus. Now if you look at the interior, you can see it's pretty special. There's a circulatory system. So we have a capillary bed. I tried to represent the arterial side of it in red and the venual side of it in blue. We have this other thing in the middle here, and that's called a lacteal. Each of these two types of structures inside the villus absorbs a different kind of digestive product. These two here don't go the same place as the other six, except five. Glucose, amino acids, the nitrogenous bases, phosphatides, and sugars all go into the circulatory system. So these ones here, I'll just move this over a tiny little bit. These ones all go into the blood. The fatty acids and glycerol, they get into these columnar epithelial cells and they get put back together to make neutral fats. Fatty acids and glycerol combine into neutral fats inside these cells, and then they get moved into the lacteal. The lacteal is actually a component of the lymphatic system. The lacteals all join together and they travel throughout the body, and eventually, after they go through lymph nodes and travel up through the neck area here, they go back into the circulatory system right about here where the jugular vein meets the subclavian vein. So the stuff eventually gets back into the circulatory system, but it has to be treated by the lymph nodes first. So now we've got our products of digestion all absorbed into the circulatory system, except these, they go into the lacteal. We have to consider, after the absorption of the food products has taken place, we have to consider what's left of the intestine. We go back and think now, at the bottom of the ileum. We've had several meters of ileum up here, and all the absorptions occurred up here. At the bottom of it we have another sphincter. This sphincter is called the Ileocecal Valve. And what it does, quite simply, is it prevents the movement of this material too quickly into the large intestine, which follows this, the large intestine or the colon. If we consider what's left in here, it used to be chyme. We've removed all of this stuff from it. The stuff that's left, we've got water in there. We've got things that couldn't be digested. And it goes past this ileocecal valve into the first part of the large intestine, which is actually the cecum, hence the name ileo, from the ileum to the cecum. Extending down from the first part of the large intestine, the colon, this being the cecum, is the appendix. The appendix, for most part, is considered a vestigial structure. It doesn't really have a function anymore, although there are some hypotheses that suggest that it does store white blood cells and it is involved in the immune system and that sort of thing. But people have had their appendixes removed. All they do is snip it off if it gets too inflamed, and they sew the little spot back together, and it doesn't do them any ill harm. So it's not really a necessary structure. It's not necessary for survival. The large intestine starts here, and it actually follows a route that goes up and around the rest of the digestive system. So it has a loop that goes up, across, and down. I'm going to erase these words here so I can show you where it goes. Oops. Up, across, down. There's another sphincter here, and after that sphincter is outside. This sphincter, I'll put the name on the other side. This sphincter here is the anal sphincter. So if you look at the large intestine, or we call it the colon as well, the cecum's here. It's got a part that goes up. It's called the ascending loop. It's got a part that goes across, called the transverse loop. It's got a part that goes down. It's called the descending loop. And then the last part of it here, a little bit more specialized for compaction, is the rectum. Now what goes on in here is the materials that are left in here have to be prepared for defecation. Defecation is simply getting rid of feces. Feces is the waste material. You can see that part in the word here. Feces is the waste material, the stuff that couldn't get digested. Most of feces is things like cellulose, which is dietary fiber. Because we can't digest that. Cellulose has the bonds between the glucose molecules. We don't produce an enzyme to take it apart. So all of the plant matter you eat contains cellulose from the cell walls. You can't digest it. It gets into the digestive system, it gets treated like everything else, but it still winds up coming out the other end. There's another very important feature of the large intestine, and that's the presence of bacteria. The bacterium that's in the large intestine is Escherichia coli. It is symbiotic relationship. I should say it has a symbiotic relationship. It's anaerobic. It doesn't require oxygen, so it's anaerobic without oxygen, and it helps us. What it does is it takes charge of this type of material here and it breaks it down even further. The Escherichia coli, the E. coli, lives off of the cellulose and the other undigested material, and it gains its energy from that. In part of that process, it releases minerals. It actually produces some vitamins. And these things, the minerals and the vitamins, are useful to us. So that's the symbiotic relationship. Because it's in our intestines, it survives well in there. It makes things that we need. So the symbiosis is a two-way street. They help each other out. One of the things that happens in all of this is it also produces gas. Methane gas. So we have gas bubbles in here sometimes. If you've ever gone for a run and you get a side stitch and you have to stop running, what's probably happened is the gas bubbles have collected in one of these two corners. And they apply pressure in here, so that's the stitch. Eventually you can get it to go away because the gas works its way through the system a little bit further. But that's what causes the side stitches when you're running. One of the things that got released all the way through the digestive system is water. Water was part of the saliva, water got released in the stomach from the mucus, water got added from the pancreas, water got added through the intestinal glands. We can't afford to lose all that water. We put tons, I can't say tons, but a lot of water into the digestive system during the process. All of that water has to be reabsorbed. And that's the main function besides defecation, is the reabsorption of water. It's not needed for hydrolytic reactions anymore, so the water can be removed from the large intestine and goes back into the blood. Along with it, it takes the minerals and the vitamins. So this material, as it passes through the large intestine, gets less liquid, because it's very liquid. It's chyme here, remember, very liquidy. It gets less liquid, less liquid, less liquid, less liquid, solid. So, and by the time it gets down here, usually it appears relatively solid, but it's not. It's still mostly water, because it would be too hard to get rid of it if it wasn't still a lot of water. In our last segment of this series, I want to summarize the whole process for you. I want to take a look at everything that happened, but I want to do it in a different manner. I want to look at it, the chemical processes, I want to look at the physical processes, I want to look at the changes in pH. You know, take the individual topics and then extract them from the different portions of the digestive system and put them together in a different format. And I think you'll find that quite useful. Thank you. Well.